I'm Charles Martini. I work in the Sutton Lab, and my project is on MRI perfusion imaging with a partial separability method. So to understand how we do MRI perfusion, we use an MRI method called arterial spin labeling. This method doesn't require any contrast agents, so there's nothing injected into the bloodstream. The signal comes from the endogenous blood. And this method quantifies perfusion using water density MRI images. In the bottom left, is a water density weighted MRI image, and that's the background signal that we would get. So first we suppress that signal because we're looking for the blood, not the background, and we use the way we suppress this is with radio frequency pulses. And then we label the blood upstream using more RF pulses. That's the tagging slab, and then that blood has the feeding arteries that go into the brain. And then we take the image, this imaging slab, and then we get the ASL image that you see on the right. So a major problem with this method currently is that it has a very low signal to noise ratio, or SNR, because the blood doesn't have that much water in it. This results in large voxel sizes and a low spatial resolution. So one thing we do to increase the signal is to take multiple images and then average them all together. So instead of taking one ASL image, we might take 40 and then average them all together to increase the accuracy of the signal. And we want to use partial separability, which is a model-based method to increase the SNR and the spatial resolution. So to think about how we do this, we have to think about different ways to sample the data. So MRI acquires data in something called the spatial frequency domain, which is called K-space. And here, this is an example of a three-dimensional K-space where you have a KZ, KY, and a KX axis. And for each KZ, we take a KX, KY spiral for each of these KZ indexes. And in the standard method where the data is fully sampled, we take all of these 32 spirals for every single time point that we average together. And a problem with this is that when we start sampling at the beginning of the time point, the signal starts to decay with a time constant T2. And so by the time we take the last spiral, the signal has decayed away. And that decreases our spatial resolution for the higher spatial frequencies. So for the PS sampling scheme, over here, we only subsample the data. So we only take 16 of the 32 samples instead of fully sampling the data. So that means that there's less T2 decay because we're acquiring the signal for less time. And in the center, we have a navigator signal where we fully sample those lower spatial frequencies through time, which is going to tell us how the big picture of how these time points change throughout the time series. So to think about how we build this into the model, just the intuition of it, you can look at the bottom figure where the intuition shows that if you know everything, then you don't need to take any measurements. And if you don't know anything, then you need to fully sample the data. So we know that the data fits a spatial temporal correlation. We know that we're collecting ASL images of the same person through time. And it's not, the images aren't just random pixels one after the other. So we know there's a very strong spatial temporal correlation. And so we can build that into the map and take advantage of that to subsample the data. And the way that we do this is by breaking down the K-space data, because S is a function of K and T, is the K-space and the time. And we decompose that into L spatial basis functions and temporal basis functions. So we get these spatial basis functions, which are C in the temporal basis functions row, and we separate the spatial basis as a function of K-space and the temporal basis as a function of time. We multiply these together, and then we take that sum of products, and this sum of products of these separated functions. It's what's called partial separability in math. And then we have the big L, which tells us how many functions to use. And we choose that by keeping that lower than the maximum value. That's what enforces the spatial temporal model. And for the row for the temporal basis, we get that from the navigator signal. So that tells us how things are changing through time. And then to find the spatial basis functions, we solve an optimization equation where we fit the spatial basis to this temporal basis. And then we also have that be consistent with the data. So this is keeping the data consistency as well as enforcing the spatial temporal model, which allows us to subsample the data. Then when we have that data in K-space, we can take the inverse Fourier transformation to get to image space. And so these are the results we have so far. We have the PS recon on the left, and on the right is the standard reconstruction. So the bottom is the axial perfusion images, where there's you can't see much of a difference in the data quality. And above that are the coronal slices, and the Z direction is in that inferior superior direction. You can see a higher sharpness in the partial separability recon compared to the standard recon, which is more blurrier in that direction. And above that, we show the frequency domain, where you can see more of a vertical axis within these case spaces, and the standard recon is more of this horizontal axis. And that vertical axis tells us that we're collecting higher signal within that within the PS recon compared to the standard. And that we have those higher frequencies that correspond to a higher sharpness in the Z direction. So for fall, our plans are to apply this PS method to perfusion fMRI, so we can see throughout the time points. We can see the functional dynamics of the brain and how they change in response to tasks with higher spatial resolution. And we also want to leverage a 7 Tesla MRI to get a higher signal to noise ratio. Current results are with a 3 Tesla. We also want to adapt this method to image flow through organoids. I'd like to thank the MRFL Lab and the NBM Fellowship for helping me with this research. Thanks. Question?
Maybe I missed this, but what's your temporal resolution that you're able to be sensitive to for flow and diffusion?
Yeah, so right now it's 4 seconds
4 seconds.
per time point. So we take an image every 4 seconds, and then we subtract those images, and that's how we get our temporal resolution. And for the PS fMRI, we have a new method where we take our navigator signal, and we find take a singular value decomposition, and we take that those left singular vectors, and we actually because with the PSF MRI, we're actually acquiring, so like for each one of these KZ indexes, we're acquiring 2 of those instead of 1, because we have control and tag pair. Then we take a subtraction, and that's how we get our perfusion image. But for the fMRI data, where we need the spatial resolution, we actually don't have a match between these 2, between pairs. So we actually only use the navigator data for that. And then we have a way to project that onto the spatial basis of the navigator, and then reconstruct those time points for the data we didn't collect. So then we can get our temporal resolution down to 4 seconds.
Could that be improved just by taking smaller fields of view, or what's your fundamental limit to that?
So I mean, with the standard method, you can take segmented data. So you can double your spatial resolution, but then also your temporal resolution is cut in half. But with the PS recon, you can subsample the data for each time point. So you're actually sampling faster as well. Yes.
Yeah, do you know how your method would compare to perfusion CT scan?
Actually, I haven't looked into that, but that would be interesting to look into.
Because clinically, perfusion CT scan, at least right now, is used a lot more frequently than perfusion MRI. I mean, there's a variety of reasons for that.
Yeah, I don't know if they can quantify the blood signal in physical units with CT. So I think that might be an advantage with MRI. Yes.
I'm sure you may have mentioned this, but for the beautiful functional MRI image that you showed, are those human brains?
Yes, these are human.
Okay. So then you wanna, if your long-term goal is to do this imaging adapted to image organoids. And organoids are small.
Mhm.
Right, compared to the human brain. So how would you, what's your strategy to improve the spatial and resolution in the very small organoids?
So, about that a little bit. So what we're thinking is to, use a small animal scanner, which is a 9.4 Tesla, and also very small coils. And potentially use a group of organoids instead of just one. And they have done MRI on organoids. Usually when they plant them in the mice, they need to be able to see through the mice to the organoids. So it has been done before. And then, one example we think of is possibly place some organoids in a plastic sort of a pipe and then flow some water through and then tag that water and see how much of it ends up within the organoids, how much passes through the organoids.
And I have the second question is, would the absence of cerebral vasculature and organoids, would that change what are you gonna looking for compared to human brain that is active?
Yeah, so what I mentioned would be for vascularized organoids.
Oh,
But,
it is a vascularized organ.
Right, but we can also think of just imaging diffusion through the organoid surface using other MRI methods. But I think with this ASL, we would want some sort of vascularization in the organoid.